Yeah. So replications always or processes always lead to some form of mutations. You have errors to everything. And so genetic mutations are derived from such. So point mutations. So the point mutations, guys, are really and truly just a change in one base pair. All right. And that really and truly results to, mutations that can have detrimental effect. And so does anyone, let me just ask this one. Does anyone know of a typical point mutation? Or an example of a typical point mutation that occurs? I'm not sure if Olivia had included it in her slides. Anyone know of a, like, it's really common, guys. It's an example of a mutation that occurs in thank you. Thank you, guys. Thank you, Brittany. So sickle cell, guys, as we remember, glutamic acid, well, at position, I think it's position 6, if I'm correct, you have glutamic acid being replaced with valine. And, when you guys learn a bit more in cell biology, you can see how this really and truly manifests. In most cases of point mutations, they are silent, meaning that you don't really have that mutation manifesting in detrimental effects. All right, but it's just a change in one base pair. All right, so in terms of the silent mutation, we're just basically seeing that you're having a difference in terms of what should have been there and now what is there. So you're coding for a different protein altogether. Remember, guys, that we read proteins in terms of the codons, which is with the 3. All right, so 1, 2, 3, 1, 2, 3, 1, 2, 3, and you get in basically a different amino acid each time. All right, but the thing is that in terms of the silent mutation now, why it goes unnoticed is that you have, I don't remember if you guys remembered from the first class where we spoke about having just I think it's what, 4 to the 6, so you have a particular limit in terms of how many base pairs would account for the naturally occurring amino acids. Remember, we have 20. All right, 20 amino acids in general. So, and that would comprise the essentials and the non-essentials. So what that means, guys, is that you would have a single amino acid at times coding for having different codons. All right. So UGU and UGC, both of those code for cysteine. So even if you have a slight point mutation, meaning that because of a different base being present, of course, by complementary base pairing, you'd have another different one or the other strand. It's a mutant form, but it codes for the same thing. So both of them are coding for cysteine. So that's not manifesting badly. Now, in the form of nonsense mutation, you basically, as I said, corresponding RNA codon no longer codes for an amino acid. This is basically now just not an amino acid that is actually there. It's a stop codon. There are specific sequences that codes for the stop codon in genetics. UGA is one. All right, and this would basically render the entire amino acid from going any further, because you encounter now a stop codon, UGA being that series that prevents the translation process from going any further. So you don't have complete synthesis of that protein in the first place. So it's a mutant form because you're not having the original protein being created. As we see here, you would have 1, 2, 3 different amino acids constituting to the protein. Here, it's just one because you encounter the stop codon too early. And so that stops the process from occurring. Another called Missence Mutation. Now, this is also something that is the same in terms of the sickle cell disease that I was saying. So it is a form of point mutation, and it is really common. It is one that you're going to basically have a replacement of a particular particular nucleotide base, and you're going nitrogenous base rather, and you will replace it with another. So just like in the form of sickle cell, I want to use that one instead of this which is put forward so that you guys can understand it better. You will basically have a different behavior in terms of how that protein is represented. So in the case of sickle cell, guys, why it is so problematic is that glutamic acid is one that allows the shape of the red blood cell to be biconcave. It's able to really and truly change its shape. So while it's traversing through your blood vessels, I mean, it can basically, it has that viability of changing the shape so it doesn't stick. The thing is now, valine is rigid, all right? It's not that easily feasible in terms of going through narrowed spaces without clotting. And that is kind of why sickle cell is something that is of great concern, because it does not allow, and then the thing is too, whenever it gets lodged, it ensues in pain. So the individual is going to be feeling a lot of pain, especially at the joint pains. It's easily lysed as well, because what happens is that sometimes when they lodge in blood vessels, over a period of time, they get lysed. And so what that means is that you have less oxygen carriers within the individual. So just remember, guys, sickle cell is a form of the Missence mutation, which is a point mutation, of course. Frameshift mutations. These now are like insertion or deletion, and it causes a disruption of the reading frame. So what you will have, guys, is that the normal DNA that would have been coded would have been deleted. All right. What would have been coding for a particular amino acid now is either not there anymore, or you insert something else. So you're not going to have the correct protein in the first place. All right. Just imagine your skin. I mean, let's say for argument's sake, I'm not sure. This is just hypothetical. I'm just throwing out a scenario out there. But let's just say, for instance, for some reason we start seeing fibrosis on our skin. Our skin just starts to get rigid. It's not allowing that nice soft texture that we are normally having. The thing with fibrotic tissue is that it's not easily stretched, it's not easily compliant either. And even let's say that happens in the heart, the myocardium. Let's say it's something that is there, it's something that is going to result in heart failure, cardiac failure, because and all of these things, guys, these are things which happen in terms of mutations. So an individual can have congenital cardiac failure because of all of these things as well. Just having some of these mutations, and that's why I see it's really important why God put these things in place. These exonuclease activities, for example, in certain enzymes that we have, because you'll always have these things coming into play at the end of the day. So you need some correcting mechanism. So the frameshift mutation, as I said, is just insertion or deletion. Well, point mutation would be different, because remember, in point mutation, you're basically replacing. All right. So in a frameshift now, you're adding or subtracting. But in so just like adding something, you have an extra. Deleting, you have one less. But in point mutation, you're replacing. So it's the same number in a sense, but only a different base being present, which would code for a completely different protein. Or well, in the case of the I think it was a nonsense, we saw where you had a stop. It just meant that you had a different protein being formed. All right. But let's see. Look at the frame shift mutation. In the shift mutation, you have a different. So we have 1, 2, 3, 4, 5, 6, 7. 1, 2, 3, 4, 5, 6, 7 as well. But you have a deletion of the. Well, it's kind of the same thing in a sense. Let me put it that way. It's kind of deleting the nucleotide just as is. However, in terms of insertion, you may have a different protein being formed. So it's kind of the same mechanism in which they operate both the point and the frame shift. I see where you're getting at now, yeah? It's kind of the same thing, but in terms of how they manifest, you will have structural changes being more evident in the frame shift mutation. I think that is the correct thing, because you're now going to have an incorrect sequence altogether, sometimes because of one. Does that make it clearer? Okay, great. Okay, all right, I get you. All right, so let's look at this amino acid sequence. So you have histidine. In the other case now, you have histidine, but you have leucine, and I don't remember this one, guys. I honestly don't remember this amino acid. But yeah, you have a different sequence now, just because they had one different base being deleted from the sequence. So what that meant? Oh, so this is it. So I was actually honest. So as I said, the point mutation meant that you had it being replaced. All right, and why this actually occurred? So there are two different entities. Let me put it that way. There are really two different entities. And why I say it is this: how amino acids are read, they are read in the codons. Remember, so it's always 1, 2, 3, 1, 2, 3, 1, 2, 3. But if you have one less, because one less base being present, you're still gonna read them in threes. It's not replaced, It's not being replaced. It's just that you have one less. It will denote a different amino acid, but you have one base shorter, which caused that mutation in the first place. In the case of the point mutation, you had a different one, but it was just due to a replacement, not a deletion or an insertion, which would allow for the codons to be read differently. So both of them, guys, are due to how it's read. But one, the point mutation is that you basically read it as a result of replacing a base. In the case of the frame shift, you read it differently because you either inserted something, which meant the threes in which you read it now. When it reaches up to the point of insertion, where you added a new base, you're going to have a different protein or different amino acid being present. So all these mutations we are recognizing, they have one commonality, and that is that you're going to get a different protein at the end of the day. Different mechanisms, but at the end of the day, kind of the same result in that you have a change in protein that is being coded for. I hope I didn't lose anyone there. I know what I said kind of sounded a bit strange, but it's just that you have different mechanisms leading to pretty much a similar result, in that you're going to have a different protein being derived at the end of the day. So the mechanisms are a bit different, but the end result is that you're not going to have the normal protein.
Yeah. 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 Yeah.